Hello, everyone. Thank you for taking your time out of ACR to join us today. We're going to get started. So I'm at the bottom. I'm Erin, and I will be your cruise director today. And today we're going to talk about whole genome cancer profiling and then introduce you to our ultrasensitive MRD testing. And we're going to present data that hasn't been shown before. So this is going to be very exciting, and stick with us. And I'm going to be inviting up, as you can see here, we have some wonderful guests who will be joining us for our fireside chat. So just a quick introduction. We are Inocras, and if you haven't heard that name before, it's a combination of innovation and tomorrow. So we're bringing innovation of the whole genome. Our goal is really through our patented protected and through our bioinformatics is to really move the needle and to bring whole genome both to the research setting and the clinical setting. We are specialized in cancer and rare disease with more than 13,000 mutations cases done so far. While we have a global footprint, as you can see here, our lab is based in San Diego, where we are both CAP accredited and CLIA certified. And then here we're showing you some of our collaborators and customers that we're currently working with. And as I mentioned, today we're going to break the talk into two parts. First, we're going to talk about Cancer Vision, which is our comprehensive whole genome screening. And we're going to actually invite up one of our collaborators to share his data with you as well. And then we're going to finish the talk talking about our new MRD Vision, or our whole genome MRD monitoring, that we have ppm levels of detection. And we're going to close it out with a fireside chat, really inviting our speakers in our panel to dive deeper and then open it up to answer your questions. So first, I want to kind of back up, and I may be preaching to the choir here a little bit about the power of whole genome. So what do you see? What do you get with whole genome? So to orientate you, the circles are actually the whole genome at the bottom. And what you see are your chromosomes in a circle. And we're showing you that targeted panels, really depending on the size of the panel, get up to less than 0.3% of the genome. So it's a little hard to see on this screen, but there are actual polka dots in there that represent variants in the targeted panel. And the picture is representing, what do we do with that information? So it's good information in that it gives you a sense of what's happening, right? You see this line drawing, you understand that it's an astronaut, there's a child inside, there's something on its visor. So you get a hint, right? There is data available in that technique. But, as we're saying here, what do you see? You're only seeing select biomarkers coverage. You're seeing a limited number of point mutations, limited number of copy variants, structural variants. It's information, but it's incomplete information. And that's why we want to transition people to a whole genome view, that complete information, as complete as we can get today. So that's the comprehensive approach. And now if you look at the child right now, it's in color, and that image on the visor, right? Now there's more information there. That image on the visor goes from just a blur to she's actually looking up at a galaxy, right? So that's what we like to think about whole genome. It's not just about information, it's about giving you that information in context. So here you're getting more point mutations, more accurate genome-wide copy number variants, more structural variants, more accurate genomic markers like TMB and HRD, which I'll talk about in a minute, and mutational signatures. So as I mentioned, we're going to talk about Cancer Vision a couple times today. And first, we're going to introduce you to how we take that whole genome and make it clinically actionable. So right now, if we look at what do you get as whole genome, right? So we take all of that data and we digest it into a truly comprehensive report, right? So here we actually use what we call targeted enhanced. So Cancer Vision has a targeted panel of 500 depth, coupled with whole genome, both for the somatic and germline. And then what we report out is we report out those somatic findings as well as select cancer-related germline findings. And we do this in a very clinically actionable timeframe in the clinical report of 14 days. Now, one of the first questions that we had when we came out with this program was, am I losing anything by moving away from a panel to whole genome? Are there challenges? So we actually did a head-to-head comparison with the Illumina TrueSight or TSO 500. And that's what you're seeing here. And to orientate you to the graph, Cancer Vision is on the y-axis and the TrueSight Oncology test is on the x-axis. And here you can see that there's a very strong correlation. But you're probably asking, what are the pink dots? So really what we wanted to show you here is beyond high concordance, we actually were able to see that there were calls that were made, but they were actually germline. Right. And I know that also one of our theories is that we can often predict germline contamination due to the variant allele frequency. But I hope this graph shows you that that's not the best approach. And really what we're showing here is that this is the power and why you want to have that germline subtraction as part of that comprehensive profiling. Another thing that's important when we think about the whole genome approach is what we can do with the power of the information that we're able to give. So to orientate you to this, the first one under targeted panels on, I guess, your left, you're seeing the traditional HRD score. And while this was a great score and moved the field forward, you can see that there is a somewhat arbitrary cutoff of what would be considered positive. Now, when you take a whole genome approach, and you add to that traditional score, and we add some proprietary whole genome based markers into our algorithm, you now see a very distinct separation between those patients who are HRD or who are HRD positive and negative. And this is important because it can reduce the misclassification of a patient and then potentially putting the patient on a wrong medication. So with that, I'm going to pass it off so that you can see some real world clinical data.
Hi, everyone. So I'm Majd from Weill Cornell Medicine. And I've been invited here to show you some real-world data about trying whole genome sequencing in a real-world setting. So what we wanted from this study and collaboration with Inocras is to demonstrate how useful whole genome sequencing is in a clinical setting. So for our collaboration, we wanted to first make sure that whole genome captures all the findings of targeted sequencing. Also, we wanted to show how much does whole genome sequencing add to our knowledge and add targets for those patients, like real world usefulness of the whole genome sequencing data. So what we did is that we selected a particular cohort of patients, which is metastatic prostate cancer patients who progressed after androgen therapy. We particularly selected this cohort of patients because those patients have no choices of treatment. So those patients, when they progress on clinical treatment, especially androgen therapy, they became what is called castrate-resistant prostate cancer, CRPC. For castrate-resistant prostate cancer, the prognosis is very bad. It's 1 to 2 years, and the patients pass away. So we wanted to see if whole genome sequencing offers more targets and more options to treat those patients. So what we did is that we selected patients prospectively diagnosed with metastatic prostate cancer, and those patients, one of our inclusion criteria was that those patients had at least one clinical testing assay. And you're gonna see it throughout my presentation. We called it TPS, which is targeted panel sequencing, and this was one of the clinically approved tests that is like TSO 500, Oncomine, Foundation 1, and Exact 1, which is whole exome sequencing. So the primary objective of this study was, as I told you earlier, to check if whole genome can validate the finding of the clinical testing, and number two, to see if whole genome sequencing can add additional targets. So first, I want to show you the results of targeted panel sequencing. So in metastatic prostate cancer patients, and those particular patients of castrate-resistant prostate cancer, we had targets in only 6 patients out of 45 patients that were included in our study. And the cancer vision, so when we did our comparison, we found that cancer vision could validate 11 out of 11 tier 1 variants. And to tell you quickly about the tier 1, tier 2 variants, this is classification followed by clinically approved molecular panels, where tier 1 are the identified variants that have strong clinical implications. Tier 2 variants are the ones that have probable clinical implications. So the cancer vision findings, the identified variants, 11 out of 11 were all validated. Out of the tier 2 variants, 68 out of the 71 variants were also validated. So we have an overall validation percentage of 96.3. And we also went further than this, and we investigated the 3 findings that were not validated on whole genome sequencing. And we find that those 3 findings have a very low variant allele frequency, and most probably where there was a probability of false positive on targeted panels. We took it a step further than this to investigate our goal number 2 for the study, which is how much whole genome sequencing will add in terms of target and in terms of overall findings. So to talk about the overall findings, which are the probable clinical implication variants and the strong clinical implication variants, we found that at least for every single patient included in our study, there is one variant with either a probable or strong clinical implication, and it varied between 1 to 10 variants of clinical implication. And we went a step further than this to investigate only the targets on whole genome sequencing. And we found that out of 45 samples that we had in our study, 24 didn't have any targets after we did whole genome sequencing. This is compared to 38 that didn't have targets by targeted panel sequencing. The good information we had here is that in our study, 21 patients on whole genome sequencing had targets, and that's compared to 5 only on targeted panel sequencing. So I show you in this graph here, you see in blue the samples with targets. So in blue, there are 5 samples that had the same targets on whole genome and targeted panel sequencing. And this is compared to additional 16, so 5 plus 16 that had targets on whole genome sequencing when whole genome sequencing was added. And the most benefit we had from performing whole genome sequencing was in terms of rearrangement and structure variants. Those structure variants were disruptive to oncogenes like BRCA1, RAD51B, or ATM, for example, and all those genes are targetable currently in prostate cancer, especially by PARP inhibitors or AKT pathway inhibitors. And one exciting notion that I must mention here, that 35.6 of the samples didn't have any targets on targeted panel sequencing, and those we identified particularly 1 to 7 unique targets on whole genome sequencing. So we see the benefit here, and we identified the benefit, and stay tuned. The manuscript is going to be published very soon.
All right, so transitioning in, and thank you so much for giving us a background of cancer vision in the real world setting. So now we're going to transition to our MRD, and it's important. Why do we spend this time going through cancer vision? And it's really important because that's actually what we use for our baseline. So now that you see how it is impactful clinically and in the research from its own right, we're now taking that information, the whole genome information that we get from cancer vision, and then we move it into the cell-free DNA monitoring. And we actually do whole genome plus whole genome. So it's whole genome at baseline. And then we take that information and we do a whole genome to see if the fingerprint is in the cell-free DNA. So that allows us to have ultra-sensitive and also a simplified workflow because we do not have any panel as part of our assay. And I'm going to walk through both the clinical data as well as the analytical validity. So this data here is our analytical validity, where we actually took 3 cell lines with known profiles and we diluted them into MASH normal to create a concentration range that modeled the real world setting and different variants for MRD testing. We did a total of 84 experiments with ppm-seq. And as you can see here in the second bullet point, some of our readouts at this particular assay validation, we did a 40x read depth. And you can see our mixed duplex rate was 33%. So that means about a third of the were read on both strands. And then we had an absolute error rate of 5.5 to 10 to the minus 7. Thank you. I think it's great too. And then as you can see here in this analytical validation, our LOD 50 was at 1 ppm and our 95 LOD was at 2 ppm. As we transitioned into our clinical assays, we looked at a simulated analytical validation because we wanted to understand how read depth would affect our limit of detection and how that would inform our clinical validity design. So as you can see here in the graph, we actually landed on 100x to get us the LOD of 1 ppm. And then we moved into our clinical validation, which is done in early lung and ovarian cancers, where we collected the tumor at baseline and then pre and post operative. We collected the cell-free DNA. And as you can see here, we had a target read depth of 100x at 10 nanograms of DNA input. And I hope you caught that our mixed duplex rate went up. So we have more being caught by the 2 strands. And then our absolute error rate went down, which is good. So we became even more accurate. And as we then moved into the next to take a big leap forward and said that we were going to do a head-to-head comparison. So we had an opportunity of this was part of an ongoing collaboration that we had that our collaborator had already run a known validated MRD assay that is used a panel technology. So here, just to orientate you to the graph, the y-axis is our MRD tumor fraction detected by our MRD, and the x-axis is the tumor fraction detected by the orthogonal testing method. And I hope this is pretty straightforward, that you can see a strong quantitative correlation. That's the line and the blue dots. But also I want to draw your attention to the green box. So here, as you can see, there's a reference line across that is a dark blue, and that is the reported limit of detection range for that orthogonal test. And as you can see, we had 2 samples that fell below that range for that test, but were actually detected by the MRD cancer vision test. And if we go and look at them a bit deeper, so you can see the previous graphs on the left, we actually did look at these over time and looked at what, sorry, the graph got a little messed, how we were able to identify. So as you can see, even though they were missed by the orthogonal test, we can see that both of these samples had a similar biological pattern to the other positive samples that we called. And then also we did run 50 blanks, and that our MRD vision test was negative for all those 50 blanks as well, really highlighting both the sensitivity and specificity of the assay. So just to close out, to wrap up before we move to the fireside chat, this is just really a table that brings everything together. So if we think about our MRD vision, what makes us different, we are whole genome to whole genome. This allows us, coupled with the ppm-seq technology, to reach limit of detections as low as 1 ppm. And then also, again, because we're simplifying the workflow, we don't rely on panels, we're able to bring this affordably and within a clinically relevant time frame. So now I'm going to invite all of our panelists up to join me for a fireside chat. Wonderful. So I'll introduce myself first because I realized I forgot to pretty much do that at the beginning. But I'm Erin. I'm the Chief Medical Science Officer here at Inocras.
I'm Young-Seok Ju, genomics co-founder of Inocras. I'm a genome scientist by trade.
I'm Majd Al-Assad. I'm a senior research fellow in precision medicine at Weill Cornell Medicine.
I'm Ariel Haimovich. I'm Vice President of Applications in Ultima Genomics.
Wonderful. So we had some questions that had come to us previously. So I'm going to ask I'm going to start the fireside chat out by asking those questions, and then we're going to open it up to all of you to ask your questions as well. So one of the first questions that we had come in was for you, Young. Why did Inocras choose to develop a whole genome MRD approach?
So MRD is the detection for detection of the tumor DNA in the plasma. And usually the concentration of the tumor level is very low in the plasma. So less than 0.1%, even some ppm level, 1 out of 1 million. So here we need the high sensitivity of the sequencing. So that means if we have more markers, then we can go more sensitive. So usually the commercially available testing, MRD testing, uses 20 or so markers. But in this case, we can use whole genome sequencing, can trace 10,000 or 100,000 of mutation from the cancer genome, and therefore we can go more sensitive here. And another one is the complexity in the lab process. So in whole genome sequencing for MRD, we don't have to use design the personalized panel. So all the lab process can be more streamlined. So in means the turnaround time can be more quicker, and also cost of the sequencing can be cheaper than the current technology. So that is why we use whole genome sequencing in the MRD testing.
So I'm actually going to bounce it right back to you and then over to Ariel. So, one of the questions we also got in this development of this assay was, why did we choose Ultima as a partner to co-develop this assay?
Yeah, it's very, very, very good question. And yeah, so the sensitivity is very important for MRD sequencing, and also specificity is very important. So usually current NGS technique, the background error rate is 10 to the minus 3 or 10 to the minus 4. So that is not sufficiently accurate for MRD testing, because we are aiming at ppm level detection. So in Ultima genome sequencing, they launched the ppm-seq. So the background error level is 10 to the minus 7 or so. So around 1,000 times more accurate than the NGS technology. So we can leverage the high accuracy of the ppm-seq technology here. So by combining our cancer vision, we can get ppm level, the LOD, limit of detection for MRD testing. Another one is the also cost efficiency. So the Ultima genomics technique is more cheaper, more economical than other NGS techniques. So we are very happy for using the Ultima genomics sequencers in our MRD the product.
Yeah, so now, Ariel, I'll kind of pass it to you from a different perspective, because we know why we wanted you in our assay for a partner with Ultima. But, what do you really feel that Ultima and the PPM-seq brings to MRD assays like ours?
Yeah, thank you. So we're, first of all, really excited to work with Inocras about this. And I think, as mentioned here, one of the key features you need for an MRD assay to be clinically useful is the sensitivity and to have very low limit of blank. It turns out that a lot of the errors you see in the percentages that were mentioned here are actually not sequencing errors. There are deaminations, DNA damage that happen on one of the DNA strands, and the sequencing actually gets them right, but then you need to be able to correct these kind of dual strand discordances. And typically this was done by adding barcodes and then having a sequencing overhead that requires a lot of sequencing to get very small effective depth of duplex sequencing. Our ppm-seq technology is one that really enables to do whole genome with the percentages shown here, 40-50% of, if you sequence 100x deep, you get 50x duplex coverage, and that is enabling both a very good specificity and your ability to find signals at the kind of ppm level.
So in ppm-seq, we do sequencing both the Watson and quick strand of the DNA fragment. So we can do the consensus calling. So the accuracy of mutations is way higher than ordinary NGS sequencing.
Yeah, that's a really good point. And just kind of thinking about it from both the researcher or clinical perspective, could you just walk us through the end-to-end workflow? How do we do this? What sample types? How does it come in? How do we process?
So already, Erin, this is presented. So we do cancer vision testing for primary tumors to trace, and we the whole genome sequencing, tumor and germline sequencing for that, and we do MRD test sequencing with whole genome by PPM-seq in Ultima Genomics, and it is very streamlined. And our technique can be applicable to all types of the solid tumors. And I believe it is a very straightforward technique for MRD for solid tumors.
Thank you. Yeah, so I'm going to ask this question to all three because I think looking at it, it does apply to everyone up here. So, I think what the question comes is, how might MRD vision be used in the research or clinical settings? So, from each of your perspectives, what is a use case? How do you see this assay being used?
So yeah, in the research, probably our product can be applied to clinical trials. So it can be a surrogate marker for the drug responsiveness of cancer. So we can use imaging or other technique or resampling to evaluate the drug sensitivity. But what we measure in MRD is the DNA from dying cells, dying tumor cells. So we can use the MRD for measuring the responsiveness of some drug in the clinical setting. So it can probably reduce the turnaround time of the clinical trial. Also, it can make more clinical trial more economical. And I think that is the one of the application for our MRD testing in the clinical trial. Any idea?
Aaron and everyone, I can give you actually a real world example of the usefulness of MRD, especially in metastatic prostate cancer. And that example is actually one of the cases we sequenced together. And I was presenting this case a few weeks ago in a research molecular tumor board. So the patient was a metastatic prostate cancer patient who received chemotherapy 3 years ago and was maintained and stable on androgen therapy for 3 years. And recently, a few months ago, like 3 months ago, the patient had a slight increase in PSA when he was followed up regularly on PSA. But this increase on PSA was within normal fluctuation of the values of PSA. And when I asked the clinician if she would act on the additional findings from whole genome sequencing that was a PTAN rearrangement, if she's gonna stop the androgen therapy and start with a new targeted therapy, she could tell me that although there's a slight increase in PSA, she still doesn't have a clue whether the patient is progressing or not. She couldn't make a decision whether the patient should change the therapy or not, especially that radiotherapy didn't show any progress in the disease. So the repeated radiological testing showed still a stable disease, although there's a slight increase in PSA. In this case, I see that the MRD testing could help because molecular progression of the disease could be detected much earlier than a PSA increase. So I really see MRD useful in those cases, and those cases happen every day in the clinic.
I think I would add 2 points. Maybe one is on the clinical setting. I think one of the very strong features we have here is that it's a quantitative measurement of the tumor fraction, and as such, if you have longitudinal samples before and after treatment, as mentioned here, you can see this kind of the burden in the blood increase or decrease. So that can be indicated for relapse, but also for response for a drug. So I think that's a very powerful use. In addition to that, on the research side, I think one of the more exciting things we see in this approach of whole genome MRD is that right now you're doing this to actually get better treatment decisions, but there's also a future in which this is basically a data generation exercise, because you're looking maybe at these 10,000 mutations that you have, but you're sequencing everything, and you're creating a huge database that will be very, very useful for research, that will be whole genome sequencing for multiple cancer patients, and that opens the door for future applications like screening, like more complicated clinical use cases. So I think it's kind of you win on both worlds. Yeah.
I'll kind of bounce it back to you. So, one of the questions we got was, what is Ultima up to, right? What research projects are you interested in? Are you working on? I mean, we very much have valued our collaboration with you, but we just love to know a little bit more about what's happening in Ultima today.
Thank you. Yeah. So we are. This is one of our major focuses is basically taking this PPM-seq approach for MRD and making it the standard of care for multiple use cases. We see a lot of interest from both clinical labs, from pharma, and our focus right now is to start generating data with this approach, again, both to show the clinical utility, but also to set the stage for future applications. So that's definitely one focus we have, and another which I think you are on the forefront is also on the target on the treatment selection side, starting to move from the specific panels to approaches that are hybrid approaches like this, when you do both kind of a target capture for the clinically relevant regions, but also whole genome that can generate both better treatment selection, but also set the stage for these future MRD assays.